So I'm going to talk to you about dissociation artifacts, I guess, or biases in 10x workflows, or basically any solid tissue. First of all, I just want to acknowledge our funding. We're fortunate enough to have funding from the Cancer Research Trust. It's a local philanthropic. We also have funding from Cancer Council WA, the Makarrata Cancer, Australian Cancer Research Foundation, NHMRC and the ARC. So walking into a pub and seeing this makes me happy. There's lots of diversity, lots of choices. That's awesome. So I have to make a choice. I choose one. Now, if I make a poor choice, what are the outcomes? Well, it's not too bad. I can just order another beer. But I could conclude that the pub or the brewer has a bad product. I could conclude that I was an inexperienced user and that I perhaps should try again. Or I can assume that I made a good choice because I haven't tried any of the other beers. Now, really, you need to try all of them to figure out whether you actually made the best decision. Now, consider what you do if the beers cost $5,000 each. This is where we're at, okay? So there's lots of choices. Warm versus cold dissociation, single cell versus single nuclei, fresh versus cryopreserved versus methanol, and there's all these fantastic papers saying this method is better than this method. What we've tried to do, however, is put these all together into one larger monster assay. I touched on this back in February in this same room, the Cell Symposium, and now we've got to the point where we've put it onto bioRxiv, so please feel free to have a look. And instead of calling it the kidney monster experiment, it's the tasting tray. So you get to try all the different variants. So we've done warm dissociation, cold dissociation, we've done nuclei, 3 variants of that, we've done bulk RNA-seq before and after dissociation. For the single cell, we've done fresh, methanol fixed and cryopreserved. All of the data was done using mouse kidneys. We're funded to do cancer, but you can't grab a tumour and cut it in half and assume that you're going to have equal cell populations on either side. Whereas with mouse kidneys, you can do this in triplicate, and you can assume that they have roughly equal complements. So the data set comprises over 77,000 single cell, 98,000 single nucleus, and currently 9 bulk RNA-seq experiments. So typically, so this is just showing some of our first single cell data. This is unlabeled mouse kidney data, single cell data. And what you can see is there's a big cluster here and these other clusters around here. And what you'd typically do, unless you're using scPred, for example, is you'd look at what marker genes are expressed in those clusters and try to annotate that cluster as cell type X, cell type Y. We developed scMatch. It's one of a family of different methods. There's other people in this room who've developed similar methods. And basically all we're doing is we're taking each individual single cell and we're doing a blast. We're comparing it against reference profiles. And then we look at the ranking of those similarities. In this case, we're using Spearman correlations. And we can either report the best match, so the top, or we can use cell ontology because all of these have been encoded with their cell ontology terms, and that means you can test at multiple levels and report the best average similarity for cell ontology. So when we apply this to our unlabeled kidney data, it performs pretty well. And this is the first iteration. This isn't the final version that we include in the paper, but I thought this looks pretty. So this is the proximal tubules. You can see B cells, you can see podocytes down here, so you can get T and NKs, etc. To show that it doesn't just work on kidney, this is on some mouse liver data that we've been generating for a collaborator. You can see we've got stellate cells, sinusoidal endothelial cells, hepatocytes, and in orange here there's some macrophages. And then lastly, this is on a mesothelioma patient. The cells in blue coming up with the mesothelial lineage. This is using cell ontology mode. You can see this big cluster here is basically a transition from macrophages down to monocytes. And this is a transition from multiple classes of T cells. So, back to the actual experiment. So, first of all, we looked at dissociation. And we just said, okay, well we can do bulk RNA seq on the dissociated material. That's cheap. So we did that. Oh, we did that. And it's highly correlated, but then there's a bunch of genes that are induced in the warm condition and some that are more highly observed in the cold conditions. And this sort of reiterates what was reported in Stephen Potter's original paper about using the cold dissociation. So it's really nice. Then we moved on to actually doing the single cell profiling. We started off with the fresh, warm versus cold. And then we were able to do differential expression analysis in each of the cell types. And what you can see is that there are some cell types where there's much stronger effect and some cell types where there's very little going on. But in general, the warm is causing more, there are more warm-specific condition genes than cold-specific genes. If we look at the genes that are commonly perturbed in at least 5 of the cell types, we can see this pattern here. And basically the genes I'm focusing on here, they're all immediate early response genes. So these cells are stressing out. But an important thing is that this stress response is cell type dependent. Some cell types are stressing out lots, like the T cells and the B cells and the mesangial cells, where the ascending loop of Henle cells, the collecting duct cells, they're quite happy. They don't care to some extent. The next thing you can look at is populations. And you can't do this with the tumour. You need to have matching replicates. So we've got triplicate kidneys for each of these. Hence we've got error bars. And what you can see here in blue, these are cell types that are more abundant in the cold dissociation conditions. And these here in red are the cell types that are more abundant in the warm dissociation conditions. And this, again, this reiterates some of what Stephen Potter's lab originally talked about. So again, replication, great. Okay, so from that first part, we conclude that cold is better than warm, okay? So with the cold data, and we do this for the warm data as well, bury it in the supplementary. We've done fresh versus methanol fixed versus cryopreservation. This ugly pie graph is showing the breakdown of cell types we see in the fresh. Other is basically all the smaller classes stuck together in one pie fraction. And drum roll, this is what we see with methanol, and this is what we see with the cryopreserved. And when I first saw this, I thought we'd stuffed up, that we made a major mistake, because all the proximal tubule cells are gone. So but we also see this in the warm dissociation. We also saw this in replicate experiments. So we went and repeated the experiment and see the same thing. The methanol fixation, there is a change in the population, or the estimated populations, but it's a much better representation of what we see in the fresh than what we see in the cryopreserved. Now, one explanation is the cryopreservation media. So we're looking at 50% fetal bovine serum, 40% RPMI, 10% DIMSO. Now, the 10x recommendation for PBMCs is 15% DIMSO, 40% FBS, 45% media. In the papers by Holger Hain and this recent paper by Patrick Palm's lab, they've gone 90%, 90%, or 10% fetal bovine serum, depending on what cell type you're working with. I want us to repeat the experiment using 90% to see if we can rescue it, but I don't know if that's going to be the solution. And also we need money to do that. I can't keep on burning money. So then for the cell types that we actually get out, we can actually say how close are they. So forget about that we've lost the major cell type from the kidney. Just let's look at differential expression. So these are genes that are upregulated or downregulated in either cryopreserved or methanol fixed relative to the fresh control. And you can generally see that there's a little bit more going on in the methanol than the cryopreserved, but the numbers of genes that are differentially expressed here are not as many as you see between the warm and the cold. The warm and the cold makes a much bigger difference than the cryopreservation or the methanol fixation, at least in this set of experiments with this set of methods. If you look at the genes that are differentially expressed, the things that are upregulated in either cryo or methanol, in the cryo you again see the stress response. You see the JUNs, you see heat shock proteins, you see FOS. And it's cell type specific like we saw before. With the methanol fixed, however, this is a signal of ambient RNA contamination. Basically, it's the same pattern across all of the different cell types. GPX3, FXD2, GPX1 and SPINK1 are massively expressed in proximal tubules, and basically we've got a dilution series across all these cell types. Okay. We also see hemoglobin transcripts here, so erythrocytes, the RNA that's present in there is actually contaminating. Okay, so that's a single cell. Now we move on to single nuclei. And we tested 3 variants of single nuclei. They're roughly based on the Frankenstein protocol, but there's some variations there. We also did bulk, and we tried to do decomposition using bseq-sc. I know there's a bunch of other methods that we could potentially try that may be better or worse, but this is just what we've used for the preprint. And what you can see in blue, this is from single cell. In grey, this is from single nucleus. So in particular for immune cells, we see a lower number of immune cells in the single nuclei, a lower number of podocytes, but we see more Lipophenolly cells, we see more endothelial cells, depending on the flavour of single nuclei prep that we use. Now, I'll just focus on the immune cells for a minute. And in particular, we're seeing depletion of leukocytes, and this has been reported before. In the Wu et al. Paper, they've talked about single nucleus over single cell, and it's nice data, but the leukocytes are gone. And this was noted by this letter to the editor from Sullivan et al. Now, in the Broad, for the single nucleus and single cell toolbox data, buried in the supplementary, there's an experiment, and correct me if I've got this wrong, where they've taken a tumour, split it into 2, and taken some for single nucleus, some for single cell, and looked at the compositions. And these are 2 solid tissues, and what you can see in black, in blue, and in yellow, these are T, NK, and B. In the cells, they're quite abundant. In the nuclei, the T cells are gone, the NKs are gone, the Bs are gone, etc. You see a similar sort of trend with the metastatic breast cancer. For CLL, where you've got a liquid phase tumour, you're not doing a dissociation anymore. The representations are very, very close. So maybe there's a competition effect happening in the solid tissue, whereas in the liquid phase, it doesn't matter anymore because everyone's already separated. And then lastly, single nuclei needs to have a nucleus. Mitotic cells don't have a nuclear membrane anymore. So in theory, you should see a cell cycle effect, and we do see this. We need better predictors, but using cell cycle scoring function of Seurat, where it classifies things either as G2M, S, and throws everything else into a bin called G1, we see a significant difference between the cell cycle phases. And we see this across all cell types. So in conclusion, workflow choices affect cell types observed, the expression levels, the quality of the cells, and we have some indication that it may also affect the ability to pass through the microfluidics workflow. There was some sort of weird effects, but when we looked at cold and warm. Future directions. I think in this field there's a desperate need for a ground truth, and we've been trying to figure out how to do this. You can't just mix cell lines because they're not going through a dissociation product. And there's a whole series of other things we can consider. There's this proprietary RNA best reagent, there's DSP, that should say RNA later, and we're already looking at hyperthermosol for some of our clinical samples. Lastly, I'd like to acknowledge the team, in particular Elena Denisenko. She's fantastic. She generated all the analyses, did all of the figures for this. Matt Jones and Belinda Go generated 95% of the data. Leanne's new postdoc, she's generated a little bit of the data. Rui developed scMatch. And the nuclear protocols were developed by Daniel Pope, Olivier Clement, and Rebecca Simmons in Ryan Lister's lab. And lastly, why am I wearing a koala? We're having the Human Genome Meeting in Perth next year in April. It's super close to Singapore, so come on down. We've got an early career researcher symposium, over 30 invited speakers, and over 40 slots for speakers from the submitted abstract. So there's lots of speaking opportunities. This is really good for ECRs. So come on down, and if you want a koala, come and see me later. Cheers.